Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT Class 12, the chapter Molecular Basis of Inheritance, DNA Fingerprinting. The preceding section 99% of base pair sequence among humans is same. So 99% of the human DNA sequence is same. Assuming the human genome 3.10 to 10 to the power of 9 base pairs, how many base pair sequence would be there differs? So how many base pairs will be differs? Mostly that almost in 3.10 to the power of 9 base pair will be get same. It was in differs as in sequence of the DNA which is make every individual their unique in their any phenotypic appearance only. So the total the genes will be they get differ. What happened? Only the physical state will be get change. One of aim the find to as a genetic differences between the two individuals or among the individual population. The sequencing of the DNA is very time would be their completely netting and expressive task. So when the total DNA will be changed, what happen? It is automatically that physical state also will be get changed as well as sequence also will get changed. Next, imagine the trying there compared the two set of the 3 into 10 to the power of 6 base pairs. How many they complete the two set of means it is in 6 point into 10 to the power of 9 base pairs. The DNA, the fingerprinting is very the quick way to that compared to as in a DNA sequence of any two individuals. So any two individual to can easily to get identified in the DNA sequence. Okay. Next, during this DNA fingerprinting, DNA fingerprinting involves identifying the difference of any the specific regions in a DNA sequence is called as any the repetitive DNA. So here every DNA sequence will be not changed. The automatically the DNA sequence will be to get changed. DNA to DNA the sequence will be to get change. Okay, now that the DNA sequence we call as a repetitive DNA because this sequences is small structure of the DNA repeated many times. So number of times to get there repeated. This repetitive DNA is separated from as in a bulk genomic DNA with a different the peak during the density, the gradient centrifugation. So in this total DNA, they completely to get differ because here they depending upon the completely density gradient centrifugation method. Next, the bulk of the DNA from major peak and their other small peaks the referred as in the satellite DNA. Some they having as what in the satellite DNA is also to get completely to get produced. Next, depending upon the base their composition, the trick of any guanine cytosine rich, in length of in a segment and the number of the repetitive units. So the number of repetitive, for example, adenine, guanine, thyme, the cytosine, the like number of repetitive units will be the get comes. The satellite DNA classified into as in many categories. Now here the satellite enter the chromosome or satellite DNA will be the get divided to as in a categories as a number of categories. The first one such as any microsatellites, mini satellites, etc. Like what in a microsatellite and then mini satellite. This sequence of normally do not code for any proteins. This mostly the satellite here the satellite the chromosomes completely here does not code for any proteins. Very important. It is not code for any proteins, but they form as a large portion of the human genome. But in this total genome, it contains as what genes. Okay. Next, after here, the sequence of the show has in a higher degree the polymorphism from different bases DNA fingerprinting. So in this total DNA base pairs, the different sequence will take place, different the polymerization also will be get takes place. Next, since the DNA is since the DNA from every tissue, like as you take as in such as blood, hair, and follicles, skin, bone, saliva, sperms, etc. So we take as an any one part of the in a human from as an individual show that in a same degree of polymorphism. So the total DNA will be the same. You take as any part that is complete their DNA will be the get same. They become as very useful identification tools. They're completely their forensic applications. It is most to get easy to get identify as any that belongs to as any which human DNA. Further polymorphisms, they're interable from as in a parent to any children. So it is also good to get help as in a their this genes will become as in a parent to children. The DNA fingerprinting is a basic their preliminary testing in the case of any dispatches. So any mistakes will be takes place. What happened to us, we can easily to get identified to as any what belongs to as any the which the children's belongs to which parent children's we can easily notice. As any polymorphism in the DNA sequence is in a basic genetic mapping, human genome was well as in a DNA fingerprinting. So genetic mapping, it is very useful to as in a human genome, human DNA fingerprinting. It is essential to understanding what the DNA polymorphism means in the simple terms. So here we are saying about the DNA fingerprinting. What is in a simple form? See here in the simple term, the polymorphism, that is in a variation at the genetic level. So in different levels, the genetic information will get coded. Arise due to the mutations. Any changes will be takes place. What happened? It is mostly takes place as an mutation. Let us we discuss as in here. Recall the different kinds of mutations and their effect through have been already they are studied about a chapter 4. So when we discuss about in that molecular base of inheritance, in that principles of inheritance, we can already learn about some mutations, right? And they are presenting the section of in this chapter. The new mutation may be arise to as in their individual, either in some somatic cells or in germ cells. So the most of the variations we are seeing as in somatic cells as well as in that in a germ cells. How the germ cells also in that in a they generated as in a gametes and sexual the reproducing organisms. So sexual reproducing they get showing as in a some changes. Am I right? Next, the germ cells the mutation does not the seriously their impair individuals ability to have any offsprings who they can be transmit the mutation can be then spread to as a completely one to another member another members of the population through what reproduction through as a sexual reproduction. Next, I'll take again the recall to definition of alleles and chapter 4. So what is alleles? What is any genes? Already we discussed in as in the chapter principles of inheritance and variation. The sequence of variations has in the traditionally has been there described as in a DNA polymorphism and more than the variant alleles means loci. So we are at a completely at locus occurs as in a human population with any the frequency completely greater than as in 0.1. Okay, now the mostly human population in the frequently greater how much 0.01. Is a simple term the inheritable the mutation it is observed as in a population high frequency that refers to as in a DNA polymorphism. So when a frequency will be increased, what happened? The frequency will be increased. That is be completely called as in a DNA polymorphism also will get increased. Now, the probability such as any variation to be the observed in a non-coding DNA sequence would be there higher. The mutational this sequence maybe not have any the completely immediate effect and impact to as an individual's reproductive ability. So if in case any changes will be takes place, what happen immediately or impactively the changes will be that takes place on the any organism. Okay, next, this mutation keep on the accumulating the generation after the generation. So and it is one of the ways the variability polymorphism. So it is complete generation to generation it comes as an equal. Next, this variety of different types of polymorphisms they are completely dragging from as in the single nucleotide changes very large scale changes. So one nucleotide will be changed, what happen automatically the total sequence will get changed. For evolution from as in the specification such as a polymorphism play very important role in that you will study in this detail in higher classes. So in this total polymorphism condition how this takes place we discuss in that in next higher classes. Next, the technique of the DNA fingerprinting was initially developed by as in then Alec Jeffreys. It is initially developed. It is used as in a satellite DNA, the probe that can be shown as in a very high degree polymorphisms. So it taken as in that high degree of DNA polymerase. It was called as a variable number of in their tandem repeats, simply called as the variable number of tandem repeats. This technique used as earlier involved as in southern blot hybridization using as the radiobudable VNTR, simply called as VNTR probes. It is including as an isolation of DNA and the digestion of the DNA, restriction endonucleases, and separation of the DNA fragments by as electrophoresis, and the transferring means the bloating process, the separated DNA fragments of synthesizing as in a membrane such as their nitrocellulose nylon, and hybridization of using label VNTR probe, and next the detection of hybrids, the DNA fragments in autoradiography, and schematic representation of the DNA fingerprinting show as in figure 5.6. So this completely VNTR they are followed in this total sequence, isolation of DNA, digestion of, this comes as in that next higher classes. What is isolation, what is in restriction enzyme separations, and electrophoresis, everything it comes as in that next. Here VNTR, it is belongs to as in a class of the satellite DNA, is refers to as in that mini satellite or small DNA sequence. They are arranged as in a tandemly. It is many copy of numbers. So one DNA sequence, small DNA sequence, you are producing as in multiple copies. The copy of number, the variable for as in a chromosome to chromosome in an individual. So chromosome to automatically they show as in some variations. The number of the repeated show as in a very high degree of polymerism. So here the completely the frequency will be get changed automatically. The number of copies also will get changed. As per result, as a size of in a VNTR, the varies in the size of 0.1 to 20 kb. So around as in 0.1 to 20 kb, it is a different type of various will be there present. Consequently, there after hybridization with VNTR, the probe, their autoradiogram gives as in a many completely bands of any the differing size. So size also here what happens, size also will get show as a variation. The bands of given characteristic the pattern for as an individual DNA. So here character will be changed and pattern also will get changed. It differ for as an individual to individual in a population, the except in case of any the monozygotic means that completely show as an the twice, right? Identicals, yeah, twins. So they get show as an twins. Here the sensitivity of in the technique was in the increased by using as a polymerase chain reaction. It will be the study about as in the chapter 9. So here when the number of copies with the help of what in a PCR, with the help of in a PCR we can develop as in many copies. Consequently, the DNA from as any the single cell enough to perform DNA fingerprinting analysis. So in the single DNA, one single cell, the container as in a DNA, that DNA sequentially it is produced as in many copies with the help of DNA fingerprinting analysis. In addition of applications of forensic science, it has much that window the application there such as here, such as the determining the complete population and genetic their diversities. The currently many the different probes are using as in a genetic in generated DNA fingerprints. So when we are using as this all, see it is in a total DNA fingerprinting, right? See it is in a total pattern of the in a DNA fingerprinting. See it is in a parental chromosome, it is a maternal chromosome. See in this DNA comes as a DNA from as an individual A, it is an individual B. So they are completely they are showing as in what in a, the number of their tendon repeats will, the number of repeats will becomes what happened, they are completely they get useful for as in to get identified to as in a that who are the this DNA belongs to as in a which humans. Okay na, this is in a part of the DNA fingerprinting. Okay na, this is okay. Remaining to discuss in next class. Thank you for watching. Please share and subscribe. Thank you.